Okay, today we're going to start chapter 3. We're doing 5 major areas today. We're going to cover 3.1. We're going to list the main points of the cell theory. We're going to cover 3.2. We're going to describe the functions of the cell membrane or plasma membrane and the structures that enable it to perform the functions. We're going to do section 3.3 and describe the process of cellular diffusion, osmosis, filtration, and explain their roles. We're going to do section 3.4, describe carrier-mediated transport and vesicular transport mechanisms and explain their functional roles. And then we're going to begin section 3.5. We're going to describe the organelles of a typical cell and indicate their specific functions. First, the cell theory. There are 4 basic concepts in the cell theory. Number 1 is cells are the building blocks of plants and animals. Cells are the smallest functioning units of life. Cells are produced through division of pre-existing cells. And finally, each cell maintains homeostasis. How do we study cells? Well, the study of cells is called cytology, and it's a combination of knowledge about cells from a microscopic study, chemistry, and physics. There are 4 microscopic techniques we use: light microscopy, electron microscopy, transmission electron microscopy, and scanning electron microscopy. The reality about cells is that cells in the body are actually very diverse, and each type is built for a specific function. When we teach you about cells, we're teaching you a typical model, but in reality, it's very different than that. Here are some typical cell examples, and you can see how diverse and different they are. I'm not going to name them all, but you can just look. Let's go to section 3.2, the cell membrane, or plasma membrane, or plasmolemma. That'll be on the test. Make sure you remember that. Remember, it contains lipids, proteins, and carbohydrates. We learned about that in chapter 2. There are 4 key functions. Okay, here we go. Number 1, it provides the cell with physical isolation from extracellular fluid. It regulates exchange with the external environment. It allows sensitivity to the environment and provides structural support. That'll be a test question. Here's a typical cell model. I'm not going to name all the parts, but if you look at that, you're able to get an overview, and these will be the parts that will be on the test. Here's a model of the cell membrane. It's a fluid mosaic model. You can see the extracellular fluid outside the cell membrane. You can see it's a bilipid layer. And you can see inside of the cell we have cytoplasm or cytosol. There's also a cytoskeleton that we'll talk more about. There are 6 major membrane proteins in the cell membrane. There are receptors, channels, carriers, enzymes, anchors, and identifiers. There are membrane lipids, and the major component is the phospholipid bilayer. There's a hydrophilic head facing the extracellular fluid, ECF, and the intracellular fluid, ICF. The hydrophilic heads are water-loving, and then the hydrophobic tails are water-rejecting. That's the fatty layer. They face each other. They're usually cholesterol molecules mixed in with the phospholipids. This table goes over the 6 major proteins. Receptor proteins are sensitive to specific extracellular materials that bind to them and trigger a change. For example, the hormone insulin. Channel proteins form a pore or a channel that permits things like water, ions, and solutes to bypass the lipid portion of the cell membrane. Carrier proteins bind and transport solutes across the cell membrane. This process may or may not require ATP. We'll learn that later on. Enzymes are chemical catalysts, and they catalyze reactions in extracellular fluid or cytosol. Anchoring proteins attach the plasma membrane to other structures and stabilize its position, give it structure. And recognition proteins identify the cell or non-self. This is very important to the immune system. Membrane carbohydrates will combine with lipids to form glycolipids and with proteins to form glycoproteins. These can function as lubricants, adhesives, receptors, and identifiers. Permeability is a property that determines what can enter the cell. There are three types of permeability. If something is impermeable, nothing can cross. If something is freely permeable, anything can cross or enter or exit. But the cell membrane is selectively permeable. Some things can enter and others cannot. Plasma membranes are selectively permeable. Now we're going to begin the types of transport there are. There are passive processes that use kinetic energy to proceed to equilibrium. And then there are active processes that use cellular energy, specifically ATP. There are two types of passive transport or passive processes: diffusion and osmosis. Osmosis is a type of diffusion, and filtration. In diffusion, molecules move from an area of high concentration to an area of low concentration or down the concentration gradient. Putting sugar in water is an example of diffusion. Lipid-soluble molecules can pass across the lipid portion of the membrane easily. Non-lipid-soluble molecules can diffuse through the channels provided, in other words, protein channels. This shows you the lipid-soluble molecules diffusing through the cell membrane, and then the protein molecules or the non-lipid soluble molecules going through the protein channels. Osmosis is a type of diffusion, and it has three characteristics. Number one is it's the diffusion of water molecules across a selectively permeable membrane. Number two, this happens with water because its selectively permeable membrane is freely permeable to water but not freely permeable to other solutes. And water flows from low solute concentration to high solute concentration. Osmotic pressure is the force of water of one solution moving into another solution toward the higher concentration of solutes until equilibrium is reached. This is an example of osmotic pressure. The water is moving to the greater solute concentration. And now you could see that the water level is different in picture number 2. However, we have equilibrium at that picture. In biology, there's something called tonicity. It's the effect of solute concentrations on the shape of the cell membrane. There are 3 examples. In an isotonic solution, one has the same concentration as the intracellular fluid, so no net movement of water occurs and the cell membrane remains intact. Second, there's a hypotonic solution. That's one whose concentration of solutes is lower than the ICF. The water will move into the cell, causing the cell to swell and perhaps lyse. In red blood cells, this is called hemolysis. Finally, a hypertonic solution is the third example. This is one whose concentration of solutes is greater than the ICF or intracellular fluid, so the water will move out, causing the shrinking or crenation of the cell. Here are the 3 examples. In picture 1, you see an isotonic solution. In picture 2, we see a hypotonic solution in the swelling of the cell. And in picture 3, we see the hypertonic solution and the shrinking of the cell or crenation of the cell. Another type of movement is filtration. It's a form of passive transport, and hydrostatic pressure forces water across the cell membrane. An example would be coffee. If a solute molecules are small enough to fit through the membrane pores, they will be carried along with the water. A tea bag is another example. Carrier-mediated transport is another form of transport. It requires membrane proteins to move substances and ions. proteins show specificity. It can't be passive, that means no ATP required, or it can't be active, and that means it's ATP dependent. Facilitated diffusion is an example of carrier-mediated transport that is not dependent on ATP. Active transport, on the other hand, is an example of an ATP-dependent type of transport. Active transport requires energy from ATP. Active transport can move substances against the concentration gradient. Ion pumps carry essential ions to one side of the membrane from another. An exchange pump is an ATP-driven counter-transport mechanism. The best example is the sodium-potassium exchange pump. This illustration shows the sodium-potassium exchange pump. Remember, it is dependent on ATP. Membrane transport moves larger amounts of materials into or out of the cell in small membrane sacs called vesicles. There are two major categories, endocytosis and exocytosis. Endocytosis is a type of membrane transport, and it usually requires cellular energy, or ATP. It packages extracellular material in vesicles at the cell surface and moves them into the cell. There are three types: receptor-mediated endocytosis, pinocytosis, which is cell drinking, and phagocytosis, which is cell eating. Here is an example of endocytosis. Exocytosis is the reverse of endocytosis, moving the material out of the cell by packaging substances in a vesicle that merges with the membrane. This requires cellular energy, and it's usually ATP. Now we're going to begin the parts of the cell. The first part that we're going to talk about is called the cytoplasm. It's found in the cell, and it contains cytosol and organelles. Inside the cell is an intracellular fluid, ICF, and it has higher potassium and lower sodium than the extracellular fluid, or ECF. It does have dissolved proteins that includes enzymes and other proteins to thicken the cytosol. Membranous organelles are membrane-enclosed organelles that are isolated from the cytosol. They allow storage and manufacturing of substances. Non-membranous organelles provide structure and tools for cells to do their work. The cytoskeleton is made up of thread-like filaments and hollow tubules made of protein, giving strength and flexibility. The most important cytoskeletal elements are the microfilaments, the intermediate filaments, and the microtubules. Next are the microvilli, and they're finger-shaped projections on the exposed surface of some cells. They provide additional surface area, and they're found in cells that transport substances from the extracellular fluid. This is an image of the microvilli and the cytoskeleton and the microfilaments.